So we're going to keep referencing this drawing that I did on the whiteboard as we add our new stuff for today. But first, I want us to look back at that graph and think about how the graph correlates to the drawing that's on the whiteboard. So first off, right here where we have that flat line, what's going on in the cell at that point? Nothing, exactly. That's when the cell is resting. So we call that the resting membrane potential. And then we said last time that right around here, something happened, right? What happens at that point where I drew the arrow? That's where we see the neuron releasing acetylcholine. Well, if the neuron is releasing acetylcholine at that exact point, what's changing with the cell? Which one? What literally changed at the sarcolemma when we released the acetylcholine just then? We opened which channels? The chemically gated ones, because acetylcholine binds it. So which one was that? What did it allow to move? Sodium and potassium. So right there where I drew the little X, we have sodium and potassium moving through our chemically gated channel that just opened up. Except who moved more again? Sodium moved more. That's exactly right. So we have more sodium moving, and it's going into or out of the cell? It's going into the cell. And so this first sodium that's coming into the cell is going to help create what effect?
Positive inside charge.
What do we call this effect that's happening right here by our acetylcholine receptor? Local depolarization. So between the dotted white line and that resting point, everything between where I drew the X and where I drew the red line, that's going to be our local depolarization. And then right there where I drew the red line, that's actually a significant point. Do you guys remember what the label was for that dotted white line last time? Oh, it's threshold. And we care about threshold, or you need to care about threshold anyway if you don't yet, because that is the point of no return. If you hit that point, you're getting an action potential every single time. And each action potential that's created is going to be the exact same strength. So if we hit threshold, the reason why that's the point of no return is because we have something new happening right at that point. Who opens there? Exactly. We have the voltage-gated sodium channels opening at that point. And so we have this flood of sodium that comes inside the cell, and that's what creates the true, full depolarization of the action potential. And notice, if we look at the very top of our curve and we compare that to where we are in the membrane potential, by the time we hit the top of the action potential, we've reversed that membrane potential. It is now more positive compared to the outside. The inside's more positive compared to the outside. That's that reversal that we've been talking about. All right, but we have a couple more changes to discuss. So right here at the top of the curve, What changes now? The sodium channels close. That's exactly right. And? Right. And so if these potassium channels are opening, that means they must be gated. What's causing them to open? The voltages. So we have other voltage-gated channels opening now. And what direction did the potassium move? Out of the cell. So through this full part of the curve, we have potassium moving out, and that's restoring our membrane potential, bringing it back to its resting point. So remember what we called that? We called that repolarization. Any questions about any of that? Maybe it's a little more clear now that we've gone over it a couple of times, I hope. All right, let's go ahead and move on to our new stuff then. So we're going to look at the excitation contraction coupling next, since that's what comes right after the depolarization. And I have a lot of words. They're mostly for your reference for later. So I'm not going to stand up here and read all of them to you. I'm going to explain it based off of a picture. But while we're here looking at the words, I do want you to make a note for yourself about the latent period, because that's something that we see in several of the chapters here in A&P. And a latent period is the period between when you have the stimulation happening, like when our neuron is releasing acetylcholine, between that and when you see the net effect, when you see the actual contraction. So it's like we've hit the skeletal muscle cell with the acetylcholine, and now it looks like nothing is happening because we're not seeing a contraction yet. But actually there are all these signals going on inside the cell because the EC coupling is happening. And I want to show you what that is. These words are for your reference for later. I want to describe it up based off of the picture. So looking at this figure, the stuff in gray at the top is the sarcolemma. So anything above my arrow is the outside of the cell, below it is the inside of the cell. And we can see if we follow the sarcolemma along that, hey, just like I drew on the whiteboard, there's this area where it dips inside the cell, our T-tubule. And if we look on either side of the T-tubule, what do we have on either side of it? We have the sarcoplasmic reticulum. Remember how it had those fat parts, the terminal cisternae, that sat on either side of the T-tubule? And so we called those three things together a triad. That arrangement's important because that way when we change stuff inside the T-tubule, we'll see a bigger effect with the sarcoplasmic reticulum. All right, so let's see exactly what's going on here with these arrows. Notice how it looks like there are these pink arrows that are moving along the sarcolemma. That's the action potential. That's the depolarization, that wave of positive charge that's moving along the sarcolemma. And notice how when it hits the T-tubule, it goes down inside the T-tubule. Well, it does that because that way it can change something inside the membrane of the T-tubule. Because the T-tubules have voltage-sensitive proteins. It's kind of like a voltage-gated channel, but it's not a channel. It doesn't open and close. Instead, this voltage-sensitive protein that we find in the membrane of the T-tubule, it's going to move. So we zap it with some change in voltage, it moves. And when it moves, it bumps into our calcium channels in the sarcoplasmic reticulum and opens them up. So it's just like hitting a button. We have this protein that's going to move and press on our calcium channels in order to force them to open up. And once they're open, Well, calcium's just an ion. It's going to move from high concentration to low concentration. So how should it move? Into or out of the sarcoplasmic reticulum? Yeah, clearly our calcium storage container better have a high concentration of calcium. So, and they show us that with the little black arrows. The calcium is being released and it's going to the cytoplasm of the cell. But we're not quite done with the EC coupling yet because we need to see where the calcium goes from there. And to understand that, we need to remind ourselves about some of those parts on the sarcomere that we were just talking about. So we already know that tropomyosin is the little rope that's in the way on actin, right? It's not allowing the myosin heads to bind right now. But with calcium, we can have calcium. That's what the little red dots are supposed to be right here. Those are calcium ions. And notice what they've done. They've attached to troponin. And when they attached to troponin, they pulled tropomyosin out of the way so that now the myosin binding sites on actin are exposed. And so now the myosin head can reach up and grab onto actin. That's kind of the final part of the EC coupling. We have to have the calcium go and bind to troponin. That way we can start the contraction. When we talk about the contraction, you're going to see this term crossbridge a fair amount. When we're talking about a crossbridge, we're talking about the myosin heads reaching up and making contact with actin. So it's like the myosin head is forming a bridge between the myosin filament and the actin filament that way. Remember that for every on switch, there's an off switch. So we have to think about what's going to help us clear out the calcium, because as long as calcium sticks around, the sarcomere is going to be able to contract because tropomyosin is being pulled out of the way. So what do you think we're going to need to get the calcium back into the sarcoplasmic reticulum? Yeah, we better have an ATP pump working. So on the little drawing, if you drew your drawing along with me, you might want to add this to yours. So besides our calcium channels that open when the voltage-sensitive protein bumped into them, we also are going to have calcium pumps that require ATP to force calcium back into the sarcoplasmic reticulum to allow the muscle cell to relax. Oh, and I added this figure because I like the fact that it shows us the sarcomere when calcium levels are low and it's relaxed. And then it shows us the calcium levels rising, calcium binding to troponin, pulling tropomyosin out of the way. And then it even shows us the final step where that cross bridge forms and now the myosin head can attach to and pull on actin. And since we've talked about all three, well, we haven't really talked about the third major step yet. There's a little more to the contraction than what I'm showing you right now. But we've hit all the points in that video that I used to introduce the contraction. So would you like to see that again to kind of picture how all this stuff is working together? Mm-hmm. Yeah? All right.
When a neuron stimulates a muscle cell, an action potential sweeps over the plasma membrane of the muscle cell.
Well, that's not quite working. Let's try that again.
When a neuron stimulates a muscle cell, an action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger a contraction. Muscle cells have an elaborate architecture that allows them to distribute calcium ions quickly throughout the cytosol. Deep tubular invaginations of the plasma membrane, called T-tubules, crisscross the cell. When the cell is stimulated, a wave of depolarization, that is, an action potential, spreads from the synapse over the plasma membrane and via the T-tubules deep into the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent sarcoplasmic reticulum, which is the major calcium store in muscle cells, thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called a myofibril, the calcium interacts with protein filaments to trigger contraction. In each contracting unit, or sarcomere, thin actin and thick myosin filaments are juxtaposed, but cannot interact in the absence of calcium. This is because myosin binding sites on the actin filaments are all covered by a rod-shaped protein called tropomyosin. A calcium-sensitive complex, called troponin, is attached to the end of each tropomyosin molecule. When calcium floods the cell, troponin binds to it, moving tropomyosin off the myosin binding sites. Opening the myosin binding site on the actin filaments allows the myosin motors to crawl along the actin, resulting in a contraction of the muscle fiber. Calcium is then quickly returned to the sarcoplasmic reticulum by the action of a calcium pump. Without calcium, myosin releases actin, and the filaments slide back to their original positions.
All right, so that's where we've gotten to so far. We just need to take a closer look at what's going on with those myosin heads to understand this whole contraction bit. And to do that, we've got to talk about the Crossbridge cycle. Remember, if it's a cycle, it happens in a circle. Where you start is eventually where you're going to end up. Okay, so there are 4 phases that are going to help us with the Crossbridge cycle, which is actually allowing the contraction. And as we talk through this, what I want you guys to think about, because otherwise it will be a little confusing, is that even though they're showing us just the one myosin head and what's going on with that, you have to keep in mind that there are others that are helping it. So that if this one releases, there's going to be another one that's up grabbing actin to pull on it while this one's relaxing. Does that make sense? So that it's kind of like the myosin heads are walking along actin and pushing it toward the middle of the sarcomere. All right, so we're going to start where the crossbridge actually forms. In order for this to happen, the ATPase, the enzyme that's part of the myosin head, has to have already worked. So notice what we've done to ATP. We've already popped off one of its phosphate groups, because remember, it's adenosine triphosphate, meaning it has how many phosphates? Exactly. So all we've done is we've popped off one of those phosphate groups, turning it into adenosine diphosphate, and then it's still holding on to that other phosphate group that it stole. After we form the cross bridge, we have the power stroke, where the myosin head literally pulls on the actin filament to pull it toward the M line, the middle of the sarcomere. And as it does this, it has to release the ADP in the phosphate group. That is really important, because that gets us ready for our next step, where the myosin head has to detach. Detachment cannot happen without new, fresh ATP. So getting rid of ADP opens up that ATP binding site on the myosin head so that a new one can come in. And when the new ATP binds, that forces the myosin head to release actin. Then the ATPase works again. It pops off a phosphate group so that we can cock the head and get it ready to start a whole new crossbridge attachment. So you need to know all those steps. You need to know what ADP and ATP are important for. You need to know when the head releases the ADP and phosphate group. And honestly, I think one of the hardest things to remember here is the fact that the ATP itself isn't actually what's allowing the myosin head to interact with actin. It's counterintuitive. ATP has to come in to force the release. And so in order to think about that, I always think about rigor mortis. What is rigor mortis?
Where the body hardens up and decay.
Yeah, after death, the body stiffens up, right? Well, that's because, think about it, if the body is dead, the cells aren't making ATP anymore, so we don't have this ATP to allow the myosin heads to release. So yeah, make sure you make a little note about that, rigor mortis. You won't forget what ATP is important for here.
Doesn't it release on its own though, after a couple of hours? Rigor mortis, it sets in and then doesn't it go away?
It will go away, but you have to remember that the proteins will start to break down.
Gotcha.
Mm-hmm.
Wait, so rigor mortis is there's too much ATP?
There's not enough. The cells aren't making new, fresh ATP. So the myosin heads get locked up on the actin. And that's where I'm going to leave us for today. I had thought about getting into some more new stuff, but I thought you guys would appreciate an extra long break before that lab test. So if you have a question about anything else, feel free to come up and see me while I break things down here. Otherwise, I'll see you down in lab around 2.